In this first Explore activity, we walked through the experiments that led to the discovery of DNA structure. Briefly, this is what we knew so far. So Gregor Mendel had done a bunch of experiments on pea plants looking at the basic laws of genetics. He knew that each individual, so we have these two pea plants, each individual contains two alleles for the same gene. Now they could have the same alleles, like be homozygous dominant, or they could have different alleles and be heterozygous. He came up with the law of segregation, which basically just said that when they go to produce gametes, one of the alleles goes to one gamete and one goes to the other. And so this one will create only big A gametes, this one could create big A and little a gametes, and then those can combine to make the offspring phenotypes. He also came up with the law of independent assortment. This one stated that if we have a second gene, let's say it's the B gene, that its segregation is totally independent of the other one. So in this particular bean plant that's heterozygous for both, you could get several different combinations of gametes. The other bit of information we knew came from a guy named Morgan, who was working on fruit flies. And he basically found that the inheritance of their eye color actually corresponded with these things called chromosomes. So now we knew that these inheritance patterns that Mendel came up with somehow reside on the chromosome. So what is a chromosome? Well, it turns out a chromosome is made up of two components, both DNA and protein, in this crazy structure where the DNA strands are wrapped around these protein balls, which are wrapped around each other, that form the chromosome. So then the question became, which part of the chromosome is the actual inheritance material? There were two candidates. The protein balls, which are made up of 20 different amino acids, so your alphabet would have 20 letters, or the DNA string, which was made up of only 4 letters. So the race was now on, and this brings us to the experiments that were done to try to figure this out. So the first experiment was the Griffith Experiment. So briefly, in the Griffith Experiment, scientists took two forms of a bacterium, S cells and R cells. Only one of them causes pneumonia, and that's the S cells. And so they noticed, if you put S cells into a mouse, the mouse dies. If you put R cells into the mouse lives. If you heat kill S cells and put them into the mouse will live. But if you heat kill S cells, combine them with R cells, and put them into the mouse dies, and we get living S cells out of it. So they knew that something from the dead bacterial cells was getting into the live bacterial cells and transforming them into these S cells. But again, this didn't solve the problem of whether it was the DNA or the protein. So then comes the Hershey and Chase experiment. In this experiment, they used a bacteriophage. A bacteriophage is made up of protein and DNA. And so they wanted to know what was getting inside the cell. And so here was their experimental setup. In this experiment, they labeled the protein coat with radioactivity so that when they separated the fluid in the cells after infection, they could see whether the protein coat entered the cell, and we see that it did not. In this one, they labeled the DNA and did the same thing, looking at the fluid in the cells to see what entered the cells, and they found that the DNA was actually entering the cells. And so at this point, it seemed probable that DNA was the inheritance material. Chargaff began to investigate DNA a little further in different organisms. So he looked at E. coli and humans, and what he found was that in E. coli, 26% of the DNA was made up of A's. Humans, 30.3% of the DNA was made up of A's. And so we know that the actual number of A's, T's, C's, and G's in the DNA seemed to make a difference in the phenotype of the organism. In addition, he found that there's always an equal percentage of A's and T's, and there's always an equal percentage of C's and G's. We now know that this is because they're actually across from each other in the strand, but he didn't know this at the time. It was a combination of the work of Franklin, Wilkins, Watson, and Crick that we finally elucidate the actual structure of DNA. And so in this case, the first thing we find is that the strands are actually double-stranded, and the strands are running anti-parallel. So in this picture, you can see that the DNA is running this direction here, it's running this direction here. So the two strands are anti-parallel. And if we put this all together, we get the final structure of DNA, which we've seen, which is a double-stranded piece that's in a helical structure with A's across from T's and C's across from G's.